Hi, my name is Tessa Pierce-Ward, and I'd like to talk to you today about Sourmash protein k-mer sketches for large systems. Sourmash is a command line and Python library for scaled minhash analyses. Minhash methods have been around for a little while, and they compress data sets into sketches composed of representative subset of their k-mers. We know that long DNA k-mer sketches can be used for distance estimation between samples. So if we have a genome A and genome B, then the sketch is here. We can compute the Jacquard index, the intersection over the union of those sketches. And as was published in 2015, 2016, the Jacquard has a relationship to average nucleotide identity. So we can get an estimate there. Sourmash actually uses a minhash variant, scaled minhash, which enables us to do both containment as well as Jacquard index. And in particular, this enables comparisons between sets of different sizes. The majority of minhash methods have focused on DNA k-mers, which are most useful for differentiating between closely related sequences. If we have a couple example 21 mers with two base pair differences, in many cases, they are synonymous substitutions that end up as the same amino acid. And in this example, these two protein submers are exactly identical. So this is just to illustrate that protein sequences allow matching across synonymous substitution. And of course, we know this. We've been using it for BLASTX, protein BLAST for quite a long time. And actually, MASH screen is the first example of using protein k-mers for some identification. We've been lately pursuing some other protein applications. In particular, we are exploring taxonomic classification. There's some challenges in this realm. Reference databases are often incomplete. Highly divergent genomes sometimes cannot be classified, or incomplete and contaminated genomes can be misclassified. And of course, our goal is to enable real-time classification to keep up with the scale of incoming data. So we've built a snake-make workflow on top of Sourmash that allows us to string a few commands together to do this. So our first step is to use Sourmash gather, which is a greedy min-set cov algorithm, to find the best genome match. So to illustrate what this does, we have a novel genome with some strain variation in there. We can pick out the best reference genome here in blue. And here I'm just kind of representing these two genomes are different genuses, but the same species. So they're a little bit different there. So we're able to get 60% match to the blue genome, and we can find an additional maybe 25% match to the other one. And of course, some is unmatched. What we can do then with the tax genome command is take the reference taxonomic lineages assigned to each of those best or matching reference genomes, and we can transfer them onto the match percentages for our novel genome. So in this case, we're getting 60% to that species A and 25% to that other reference genome species B. But because these are non-overlapping matched regions, or match percentages, we can actually sum that to get the percent match at the least common ancestor for these two. So in this case, genus X has an 85% match. So what we're using this for then is choosing a percentage or a threshold for when we consider sufficient query match, and we can assign that classification that we get to our novel genome. Okay, so how well does this work? We decided to do first a comparison with GTDB TK, and we started out with a benchmark set of 1,000 human gut metagenome assembled genomes from Magnify, and these are all likely to have similar genomes available in reference databases. We found exact lineage matches for about 95% when using the full GTDB recent release. And the additional around 4% of it actually all matched at the genus level. So we think it's doing pretty well, even though we're using currently a very naive classification threshold of 10% of the query, and we need to look into this a little bit further. Extending this to an environmental set of samples, we took 885 genomes from the Tara Oceans, from the Delmont paper. I think neither of them really classified all of them, but they got pretty close. And here we had exact matches at about 66%, and some match at a higher lineage rank at 33%. And if we break those down, most of those lineage matches happen at the genus level. So we got the exact same genus level classification, but a different species was selected as the exact classification. So we need to look into those differences there. But it's promising because this is a really lightweight whole k-mer or whole proteome k-mer based classification, which means it's likely going to be a little more robust to incomplete and contaminated genomes. And then, of course, because it's k-mer based, it offers near real-time classification. And with that, I'd like to thank you so much for listening. And please do find us on Gitter, GitHub, and everywhere else on the internet for questions and contributions. Thank you.